Welcome to my channel Adhyan with Flow. So let's start our Calvin cycle. This is the part of dark reaction. In our previous video we already discussed about light reactions. Light reaction, where they occur? They occur thylakoid membrane. Next we already discussed photosystem. Yes, we already discussed photosystems, photosystem second and photosystem first. We already discussed cyclic and non-cyclic photophosphorylation. Photophosphorylation, we already discussed all things. And then last thing is chemoosmosis hypothesis. We discussed everything. This all is phenomena is occur in light reaction and the thylakoid membrane. Now this term to dark reaction, they occur on stroma. Look at here, this is the chloroplast, double membranous structure, and this is the set of thylakoids. Here light reaction is occur. Stroma lamellae, here is light reaction is occur. Here is light reaction is occur. But the remaining part is, look at here, this is the stroma part. Here dark reaction is occur. Dark reaction does not mean they occurs in dark, no. They occur in stroma in the daytime. Let's start with Calvin cycle. So Calvin cycle divided into 3 steps. Carboxylation, yes guys, carboxylation. Second is reduction and third is regeneration. We were discussing about this 3 steps. First, carboxylation. Let's start. Our first carboxylation. Carboxylation means addition of CO2. Let's start. What happened in carboxylation step? This is the ribulose 1,5 phosphate. Look at here guys. Here is the importance of one enzyme. I'm explaining here Rubisco. This is the enzyme. What is the importance of this enzyme? Their affinity towards CO2 as well as oxygen also. What the mean of affinity? Rubisco bind with CO2 and Rubisco also bind with oxygen. So from the atmosphere, CO2 is enter in the stroma part. So CO2 bind with Rubisco. So CO2 bind with here, CO2 bind with Rubisco and change ribulose 1,5 phosphate to 3-glycerate. I'm repeating again. From atmosphere, CO2 come inside. They bind with Rubisco enzyme. They have 2 kind of function. They have carboxylase activity as well as, look at here guys, here also write up carboxylase as well as oxygenase. They bind with CO2 as well as O2. So after binding, this conversion is occur. Conversion. RUBP change into 3-phosphoglycerate. So RUBP, when RUBP converts into 3-phosphoglycerate, this mechanism is called it as a carboxylation step. In carboxylation step, Rubisco convert RUBP into 3-phosphoglycerate. Remember this thing. Rubisco, what they do? They take CO2 from atmosphere and they convert RUBP into 3-phosphoglycerate. This term is clear. RUBP change into this. Clear guys? Clear? Very clear. Now after reduction process, 3-phosphoglycerate, they change into triose phosphate. During this conversion, ATP and NADPH, these are the energy form. Where we get this energy? You know na, from light energy, non-cyclic and cyclic pathway, they produce ATP and NADPH. So here there is a role of these. They break down into less energy form. They converge ADP and ADPH. This form to this form means energy is going. Here triose phosphate give their one carbon, and this one carbon make sucrose, starch, glucose, means food formation start. Then triose phosphate going to their back to their normal compound RUBP. This process is regeneration, and again we lost one ATP. This is the diagram of your book. This is the whole mechanism of this is the Calvin cycle of your book. This is the Calvin cycle of your book, guys. Now I am going deeply, guys. I am now going deeply. This is not an exam forming area. This is so simple, guys. Examination in your competitive examination, this is less important, guys. Now I'm telling you some depth. Whatever I tell you, these are competitive zone. They will ask in examination, your competitive examination. According to your 10th, 12th class, 11th class, this is enough. But according to competitive exam, this is not enough. I'm explaining you in detail. RUBP, guys. This product is a 5 carbon product. I'm drawing here. This is the 5 carbon product. This is the 1 carbon, this is the 2 carbon, 3, 4 and 5. Are you clear with this term? This is the 5 carbon product. I hope this word is clear. That RUBP, ribulose 1,5-bis phosphate is 5 carbon product. Is it clear? Now this step is clear. Okay. Now we have Rubisco enzyme. Rubisco enzyme bind with CO2 and converge 5 carbon to 6. Look at me. Rubisco enzyme bind with CO2. They converge. This form ribulose 1,5-bis phosphate into 3-phosphoglycerate. This form. At the position of 1, here is 1 phosphate is added. Here the location of 5, here another phosphate group is added. At the position of 3, here, look at me, guys. 1 carbon, 2 carbon, 3 carbon. 1 carbon, 2 carbon and 3 carbon. Here, at the carbon, third, they contain phosphate group. How 5 carbon convert into 6 carbon? Because 1 carbon comes from atmosphere. 1 carbon comes for atmosphere. So they add on. So 6 carbon converted into 2 molecules of 3 carbon compound. Now this thing is now clear, guys. Now this thing is clear. Very clear, guys. Very clear. Look at here, guys. We found these products. We found 2 molecules of 3-phosphoglycerate. Now this is the term for reduction process. We reduce some energy, and this energy is coming from where to where? This energy comes from light reaction by the cyclic and non-cyclic photophosphorylation mechanism. In the light reaction, their main role is what? They produce ATP and NADPH. ATP produced in thylakoid membrane and NADPH produced in stroma. I already discussed all the things. Now here this ATP and this NADPH participate. Here this ATP and NADPH participate, and they converge into ADP and NADPH. High energy form give their energy and change into less energy form. Now this thing again going toward the light reaction, and they take participation of ATP formation and NADPH. Now this term is clear. Where ATP is coming, where to where, and where this is less energy form goes back to take some more energy. Clear, guys? So during reduction process, this form will change into their triose phosphate form. Triose phosphate form. But they release their one carbon. They release their one carbon for food formation. For food formation, they contribute their one carbon, and this is remaining is 5 carbon compound, and they going back to their normal. So here saturate this 5 carbon again, this 5 carbon. Look at here, guys. Now clear, balance, reaction is balanced. Now reaction is balanced. Now tell me, we get one carbon molecule. We get one carbon molecule. But we need 6 carbon molecule for forming our glucose. C6 na, C6H12O6. Here is a one carbon, that's it. Here is only one carbon. So this phenomena occurs 6 times. This Calvin cycle rotate 6 times, then produce one molecule of glucose, guys. So food making process is very hard to plant. So 6 times cycle they going to produce and form. In one cycle they remove one carbon. Again this cycle rotate and come, they again release one carbon. Again this cycle rotate and gives one carbon. Again this cycle rotate and gives one. So they give 6 carbon. Then one glucose molecule will form C6H12O6 because we require 6 molecule of carbon. Let's talk about ATP calculation. I'm writing here, guys. ATP and NADPH calculation, guys. For one carbon fixation, we require here 2 ATP and here 2 NADPH and here 1 ATP. I'm repeating, for one carbon we need, for one carbon fixation we need 2 ATP from reduction and 1 ATP from regeneration. And for one carbon CO2 fixation we need 2 ATP NADPH, but in regeneration we does not require any kind of 2 NADPH. This is the scenario of one carbon, but we need 6 carbon na, guys, but we need 6 carbon. So what we do now, we rotate, we want to fix 6 CO2, we want to fix 6 CO2, we need 6 carbon, so we have to rotate this cycle 6 times. So here 3 into 6 and 2 into 6. Look at here na, 2 plus 1 3, and this is the 2. We want to rotate this cycle 6 times because we need 6 carbon. So calculation is 18 ATP and here is 12 NADPH. So Calvin cycle form food. Here sucrose, here starch. So these are 6 carbon compounds. We need 6 carbon. For making the one molecule of food. We need 6 carbon for making the one molecule of food. So this cycle performance happen in 6 times. 6 times this Calvin cycle performed, and then we get one molecule of glucose, sucrose or starch. So overall calculation is we need 80 ATP and 12 NADPH for making one glucose molecule. So that's all for now. That's all for your Calvin cycle. In our next video, we will cover Hatch and cyclic pathway and other kind of pathway. Hatch and slack. Look at here, guys. In our next pathway, we will discuss about C4 pathway. In this pathway, we discuss about Crane's anatomy. And this is the pathway of Calvin cycle. Because Calvin discovered this, so name goes to Calvin. First step is carboxylation. We need carbon dioxide. In reduction, we reduce some energy, and regeneration, we also reduce some energy. After reduction, we get one molecule of carbon, and this one carbon here, this one carbon contribute in glucose formation. So that's all for now. We will meet in our next video. If you still not subscribe my channel, then please guys support me. Subscribe my channel. Thank you. Keep smiling and revise. Thank you.